Welcome to part 2 of how yeast ferments. This video will expand on the content of part 1, where carbon dioxide formation was explained. This video will expand on that topic by covering ethanol generation. Fundamentally, fermentation is the conversion of a sugar solution into its two key derivative compounds, carbon dioxide and ethanol. Assuming the precursor steps covered in video 1, the process of generating ethanol sits between the TCA cycle and glycolysis. The TCA cycle begins here with the assumption glycolysis has occurred and the reaction is contained by an infinitely large vessel with unlimited sugar-rich water. The glucose is broken down into 2 units of pyruvate. This releases 2 hydrogen ions into the local environment. The pyruvate and associated carbon dioxide is dissolved into the surrounding water and is assumed to have no ongoing effect to the fermentation. The yeast then shuttles the pyruvate further into one of the 2 possible pathways. The first is the TCA cycle, where energy in the form of ATP is generated and hydrogen ions are separated. The second is the ethanol production pathway. If it instead enters the ethanol pathway, pyruvate undergoes several changes, the first being a conversion to acetaldehyde or acetyl-CoA. The first step involves an enzyme called pyruvate decarboxylase, which strips the highlighted molecules from the pyruvate. This produces acetaldehyde. This is important in several chemical transformations and biological processes. It is not only an ethanol precursor molecule, it precedes acetic acid, commonly known as white vinegar concentrate, methyl alcohol, and more. Another enzyme then breaks the oxygen double bond, opening the carbon molecule to accept a hydrogen ion. This forms the ethanol compound. So far, a number of factors have been ignored. This includes a number of rate-limiting steps. Rate-limiting steps are factors which control how much ethanol can be produced in this example. These are a few of the possible limitations in production. The first is rather obvious, the amount of sugar in solution. For every mole of sugar used, 2 moles of ethanol and carbon dioxide are made. Therefore, 1 kilogram of sugar produces approximately 1/4 kilogram of both carbon dioxide and ethanol. In theory, this can be measured by the weight lost in the wash as carbon dioxide leaves through the airlock. This measurement is further limited by other factors, which may also confound such measurement. The second is volume. A large bottle will have less capacity than a fermenter which has less than a commercial plant. This will mean there is less sugar overall, but also that any volume, concentration, cofactor, minimum or maximum capacities will be reached that much sooner. Third and final is the toxicity of the wash. As seen here, the yeast has an advertised tolerance of 20% alcohol by volume. This means that a theoretical batch in a 5-liter container, a small batch, will yield 1 liter of alcohol. However, as this graph shows, that is not the case, with a 5-liter wash yielding 0.4 liters of alcohol. Whilst the 5-liter wash yields 0.4 liters of alcohol, the 20-liter or big batch produces 3.6 liters of alcohol. This is where the effect of maximum and minimum concentrations is demonstrated. Similarly seen here is the capacity for carbon dioxide production. This has been a brief and limited introduction to how yeast produces ethanol in a home brew. The topics covered here will be expanded on in the future. Thank you for watching, and please post any questions, suggestions, or comments below.